All right, we're going to go ahead and move on. We'll do another bit of material, and then I'll give you plenty more time to work on it again, okay? So we've talked through the idea of linear projections of sugars, right? And those Fisher projections genuinely are the easiest way to visualize sugars, right? So if you can get used to drawing them, it's a fast, easy way of recognizing sugars. If you're not good with Fisher projections, at least for the context of sugars, I encourage you to get good at them because it just is the easiest. Okay, but as it turns out, sugars aren't just present as in the linear form. Nearly all sugars, not all sugars, but nearly all carbohydrates can also form a stable ring structure. Okay, and this happens through one of the hydroxyl groups, usually the very last one, like furthest away from the carbonyl, usually, not always, unless I tell you this, it'll be. So basically the one that we use to define whether it's a D or L sugar, okay? This hydroxyl group will serve as a nucleophile. It will attack at the carbonyl, and that forms a covalent bond that creates a ring, okay? So we now have this covalent bond between this alcohol group, this oxygen, and this carbonyl, okay? And it ends up forming this, in this case, a 6-membered ring, or it could be a 5-membered ring, okay? And we name these ring forms based on the common organic molecule that looks like, and sorry, that's hiding it, but a 6-membered ring that contains an oxygen is pyran, and a 5-membered ring that contains an oxygen is furan. So we call them pyranoses or furanoses. Quite frankly, I don't care if you remember that. I just wanted to tell you, okay? If you see something named pyranose, it's a 6-membered ring sugar, okay? What I want you to be able to do and be able to get good at is to convert something from the linear Fisher projection into its cyclical form, okay? And there's some tricks to it. First things first, being able to quickly recognize whether it's a 5-membered or a 6-membered ring. We'll never see anything bigger. It'll always be 5 members or 6 members, okay? So being able to quickly draw out these structures, the base structure will help a lot, okay? So let's first do, let's walk through a hexose that makes a pyranose, okay? So like glucose. So the idea here is very quickly being able to draw out this base pyranose structure, 6-membered ring with an oxygen in one position. And if you get good at drawing it, and if you learn to draw it where the oxygen is in the top right corner and it's set up like a nice clear hexagon, I think it makes it a lot easier. Okay? So you draw out your base 6-membered ring. One of the positions is occupied by an oxygen. This oxygen is the oxygen on this that came from the nucleophilic attack by that hydroxyl group. Okay, so in this case, it's on carbon 5. Okay, so what I would encourage you to do is to immediately number this now. Okay, this is the oxygen that was part of the D carbon, carbon 5. So this is carbon number 5. This is that CO bond. This bond between this oxygen and this carbon right here is this bond that we're showing forming right here, right? This the red oxygen to the blue carbon, which means this carbon right here is carbon number 1. Okay, and we give this one a spec. This is carbon 1, which means this is carbon 2, carbon 3, carbon 4, carbon 5, and then in this case we still have one more carbon, carbon 6. So we can just show this as going up, and I usually show it pointing off to the left. But there's no stereochemistry here, so show it however the hell you want. Okay? Some people would write it CH2OH. That's fine too. I just need to make sure you understand that there is a 6th carbon there. Okay? So we started off, we drew out our general pyranose structure, we numbered the carbons, and we make sure that we have all of our carbons drawn here. Okay? Now comes the trickier part. The trickier part is getting the orientation right. Okay? But if you remember, so take your Fisher projection, your linear form, and rotate it so that you're rotating it clockwise. Okay? The way that the alcohol groups point as you rotate it to the right is the correct orientation in your cyclical form. So here for glucose, we rotate it this way. Carbon 2 was pointing right, and when you rotate it, things that point right are now pointing down. So the alcohol group on carbon 2 points down. The alcohol group on carbon 3 is pointing left. When you rotate it clockwise, that alcohol group now points up. So the theme is things that point right, point down, things that point left, point up. So we have down on carbon 2, up on carbon 3. Carbon 4, it points to the right, so it points down. So down on carbon 4. And then carbon 5, we've already taken care of everything. That's the hardest part, is getting this orientation right. If you can remember the base structure of a pyranose, draw a 6-membered ring, draw a hexagon, make one of the positions oxygen, make sure it's the top right position. Number your carbons. Make sure you have all the carbons drawn. Sometimes, usually you have at least one carbon hanging out of the ring. Okay, and then in your Fisher projection, if something points right, it's going to point down on your ring structure. So on carbon 2, it's down, carbon 3, it's up, carbon 4, it's down. This is glucose, and specifically, this is D-glucose. Okay, we won't talk about how this changes if it's L-glucose, because we don't care. Right? L-glucose is not biologically relevant. So this is how to go from a Fisher projection to a cyclical projection of our sugars. Okay, at least things that are 6-membered rings. Okay, a similar theme goes for things that end up being furanoses, 5-membered rings. Okay, here we first have to figure out that it actually is a 5-membered ring. Okay, so the trick here is to also take the last chiral carbon. We're counting this oxygen because this oxygen is part of the ring, right? This is atom number 1, and it's going to make a bond with the carbonyl carbon, in this case C2. So the alcohol, the oxygen is 1 atom, C5 is 2, C4 is 3, C3 is 4, and C2 is 5, right? So being able to visualize this ends up being a 5-membered ring. 1, 2, 3, 4, 5. This is the bond that forms that closes the ring. So it's going to be a 5-membered ring. Now draw out your furanose structure. And if you're used to drawing nucleic acids, this should look familiar. All right, this is the base structure of ribose. Ribose is a furanose. Okay, let's follow the same steps. Once you draw out the base structure of your furanose, we now need to number them. Okay, so carbon 5 is attached to the hydroxyl group. So this position right here, this bond was that original CO bond. So this is carbon 5 right here. This is the new bond that formed. This is carbon 1. So 1, 2, 3, whoops, what did I do? Sorry, this is carbon 2. 2, 3, 4, 5. Okay, we've only labeled 4 carbons despite the fact this is a hexose. So what we need to recognize is that on carbon 2, we have another CH2OH. And on carbon 5, we have a CH2OH. So in this furanose, we have to have a CH2 OH coming off of C2. That's carbon 1. And right now, it doesn't matter which orientation you have it. Do it whichever way you want. On C5, we have C6 coming up. And this does matter. Make sure it points up. Okay, so for both our pyranose and our furanose, carbon 1, make sure it points up. Sorry, carbon 6. I'm getting all backwards right now. So this is carbon 6. Okay, so we have all of our carbons written. They're all numbered. Now the only thing left is to put in the position of the alcohol groups that we're not already showing, right? We've already taken care of 3 of them. We have the alcohol group on C6, we have the alcohol group on C1, and we have the alcohol group that was part of C5, which is now part of the ring. So it's really just C3 and C4 that are left. Same thing goes as we did with glucose. We're going to rotate this clockwise. Carbon 3 is pointed left, so on carbon 3 it points up. On carbon 4 it's pointing right, so it points down. So fructose has alcohol group pointing up at this position, alcohol group pointing down at this position. In the Fisher projection, if alcohol groups point right, they point down in the cyclical form. If they point left, they point up. And that will always be true for D sugars if you draw them with the same cyclical pattern that we see traditional for furanoses and pyranoses, with the oxygen in the top position for a furanose, the top right position for the pyranoses. Does that make sense? Any questions? Honestly, I think truly this is probably the hardest part about carbohydrates, is getting these structures right, translating it from the Fisher projection to the cyclical projection. Okay, other things that are relevant about the cyclical form is it's intentionally drawn here as equilibrium, right? The idea is that this linear structure is in equilibrium with the nonlinear structure. And this hydroxyl group can attack in two ways, both of which are at equilibrium. It can kind of like, so think about in our orgo, like you learn like backside attack or frontside attack, right? So the same thing can happen here. This hydroxyl group can attack from the top or from the bottom, and that impacts the orientation of the alcohol group on carbon 1. And notice that I did not, in both of these, I have not written the alcohol that was like this oxygen that was part of the carbonyl. I haven't shown its position yet on either of these, okay? Because it depends on how this ring closure happens. If this oxygen attacks from the top side, it pushes this CO bond down, so we would show it pointing down. If the oxygen attacked from the bottom side, then this would point up. Okay, and both of them are perfectly legitimate structures, but we name them differently. If this alcohol group points up, it's beta. If it points down, it's alpha. Okay, so the beta configuration is this alcohol group pointing up. The alpha is this alcohol group pointing down. Okay, and I reinforce that on the next slide. So here I drew it down. So this we would call alpha D-glucose, because the hydroxyl group on position number 1 points down. Over here, I'm going to show this one pointing up. This would be beta D fructose. Okay, the orientation of the alcohol group on that, we call it the anomeric carbon. The carbon that was the carbonyl impacts a lot of things. Okay, and this actually does become very relevant in terms of like the chemistry of these sugars. All right, other things I want to point out here is that this bond that forms, these are called hemiacetals or hemiketals. For the organic chemists in the room that care about this stuff, these are called hemiacetals or hemiketals. The reason I point that out is because you might hear these words thrown around. The general idea is that a hemiacetal is the functional group that forms when an alcohol condenses with an aldehyde. A hemiketal is the functional group that forms when an alcohol condenses with a ketone. Okay, and it's at equilibrium. In fact, in glucose, this tends to be at about 35% of the time, about 65% of the time. You don't have to care about that. But what I want to really reinforce is that both of these structures are possible and stable and always at equilibrium. Okay, so again, the orientation of this alcohol impacts how we name it. Okay, alpha, the alcohol group points down. Beta, the alcohol group points up. Okay, for 6-membered rings especially, and I really want to stress this for 6-membered rings, because this is where I'm going to really ask you to think through it. For 6-membered rings, the beta position is always more stable. Okay, always. And the reason for that is that if we think about our chair configuration, right? 6-membered rings always have a boat or a chair configuration. The chair configuration is more stable. And hopefully you remember from orgo that when everything is in an equatorial position, that's when things are most stable, right? So here, the alcohol group in the beta configuration, when it's pointing up, is stable or is positioned in the equatorial position, which makes it more stable. Okay, so the way that I remember it, it's cheesy as hell, but it's always better to be up. Okay, so the beta is the more stable configuration. Any questions about that? All right, so with that, you have enough to work through most of the rest of your activity. So I'll give you about another 15 minutes, and then we'll